Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CET question paper for the subject Biochemistry conducted in the year 2024. This is part 1 of the explanation. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Biochemistry for the same examination, you can go to my channel Omni Bioentrance. In the playlist C for CUET, there you will find. Coming to the first question. How much of protein content makes an E. coli cell in percentage of the total weight of the cell? So they have asked what is the protein content of an E. coli cell. So E. coli cell, you all know it is gram-negative bacteria and it has comparatively high protein content. So the answer is it is 50% of the total weight of the cell. Then next one. The science of collecting small molecules in a given cell under a specific set of conditions is called. So these small molecules they can include sugars, amino acids, nucleotides, lipids, hormones and various other small molecules. So all these, if you see, these small molecules, they are nothing but your metabolites present in cell tissue under various specific conditions. Okay, so the science of collecting these small molecules in a cell under given set of condition is nothing but metabolomics. So proteomics, it is the study of proteins and their interactions in the cell or an organism. Transcriptomics, it deals with RNA transcripts. Lipidomics, it deals with lipids and their metabolism in the cell. Then next, pair of stereoisomers that are not mirror images of each other are called diastereomers. They are not mirror images of each other. Whereas enantiomers, they are mirror image of each other. Then chiral center, it is the central, usually it is the carbon atom or any central atom which has 4 different groups attached to it or different groups attached to it. If it is carbon, it is 4. Then racemic mixture, it is a mixture of equal amount of 2 enantiomers, that is racemic mixture. Then next one. In 2014, scientists from Scripps Research Institute, California announced the development of 6-base DNA. So naturally our DNA, it is 4 base. So these scientists, they have developed 6-base DNA. Any new base pair that they added to the standard 4-base DNA was called. Okay, so what is the new base pair that they have added to the standard 4-base, that is ATGC DNA? So that was called as X and Y. And these base pairs, they mimic the structure and properties of the natural base pairs. Okay, and they could be replicated by recognized and replicated by DNA polymerases as well. So that is what they have developed. Then how much percentage of the human genome is represented by the exome? It is 1%. Then next one. The most abundant monosaccharide in nature, it is glucose. It is a monosaccharide, whereas maltose, it is a disaccharide, lactose again a disaccharide, and amylose is a polysaccharide. The question is for monosaccharide. So if you do the elimination technique, you can get the answer, it is glucose. Then next one. From the evolutionary standpoint, extreme thermophiles are more closely connected with which one of the following? So they are more closely connected with the methanogens. These extreme thermophiles and these methanogens, they belong to Archaea domain. Okay. And they are from evolutionary point, the extreme thermophiles are closely related to methanogens. Then sulfur bacteria are example of which of the following? They are lithotrophs. So lithotrophs, they use inorganic compound as their source of energy. And sulfur bacteria, they use sulfur compounds such as hydrogen sulfide, sulfur dioxide as energy source. And they oxidize these compounds releasing sulfate ions. Then organotrophs, they use organic compounds as energy source. Autotrophs, they produce their own food. And phototrophs, they use light as a source of energy. Then which of the following has the highest heat of vaporization? So the heat of vaporization, it is the amount of energy required to change a substance from its liquid state to the gaseous state. That is called as heat of vaporization. And the highest heat of vaporization is for water because it has strong hydrogen bonds. And you require more energy to break these bonds. So it has got the highest heat of vaporization. Then next one. From the following data, identify the correct pH value of sea water. It is 7.8. Answer is option 3. Then next one. At 25 degrees, the degree of ionization of pure water at equilibrium is. So molarity of pure water, we know it is 55.5. So degree of ionization, it is given by concentration of ionized water or concentration of H plus ions. The concentration of H plus ions, you all know it is, if you know the ionization of water Kw value, it is 1 into 10 raised to minus 14. That is the product of the concentration of H plus ions and OH minus ions. So here it is concentration of ionized water or concentration of H plus ions, that will be 1 in 10 raised to minus 7 divided by concentration of unionized water, that is 55.5. So when you divide, you will get it as 1.8 into 10 raised to minus 9, that is approximately 2 into 10 raised to minus 9. Here it might be a printing mistake. They have given 2 per 10 raised to 9. Okay, it is 2 per 10 raised to 9 molecules. Then it is correct. So answer is option 3. Then hungry bone syndrome is associated with. It is associated with hyperparathyroidism. So what is hyperparathyroidism? It is a condition where parathyroid glands, they produce excess amount of parathyroid hormone, and this leads to elevated levels of calcium in the blood. So because of this hyperparathyroidism, the treatment is they will be surgical removal of the parathyroid gland. Now because of hyperparathyroidism, the bones in the body were accustomed to high levels of parathyroid hormone and calcium. Now after surgical removal of this parathyroid hormone, they experience a sudden drop in this calcium levels, and this will trigger uptake of calcium by the bones. Okay. So this condition is called as hungry bone syndrome because the bone is hungry for calcium. Then diabetes mellitus, it is characterized by high blood sugar levels. Then sodium is not related to hungry bone syndrome, and vitamin B also not related to hungry bone syndrome. Then next one, they have matched the following. They have given the functional group and the description. So hydroxyl group, it is present in polar compounds such as polar amino acids. You will have hydroxyl group in them. So A will go for 3. If you see the options, you can see A3 is there only in 1. So naturally one must be your answer, but we will see the others also. Then carboxyl group, it is present in fatty acids. Amino groups, okay, in proteins, 2C2, and phosphate, DNA, phosphodiester backbone. So D1. So answer will be A3, B4, C2 and D1. Then next one. In cytosol, the product yield from the oxidation of one molecule of glucose is. So one molecule of glucose, you all know, glucose gets splits up into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate in the glucose pathway. So 3 carbon compounds they are forming. Now in the end, when these 3 carbon compounds undergo the further step of glycolysis, you get pyruvate. Okay, now since glucose is a 6 molecule carbon compound, it will yield 2 molecules of pyruvate. And in the glycolysis step, you have got NADH being formed. So there will be 2 NADH and 2 ATP. So the answer will be 2 pyruvate, 2 NADH and 2 ATP. Then next one, hypokalemia. Okay, what is it? So hypokalemia, it is potassium levels in the blood are normal, are lower than the usual. So A will be 4. A4 is seen in option 1 only. So 1 must be your answer. Okay, we will see. What is hypocapnia? Hypocapnia, it is where carbon dioxide levels in the blood are lower than the normal condition. So B2. Then pseudoriferous glands. They are known as sweat glands and they are found in the skin. So C will be 1. And Sertoli cells, they are nurse cells found in seminiferous tubules of the testis, and their function is to provide nourishment and support for the developing sperm. They are nurse cells. Okay, so it is involved in spermatogenesis. So D3. So answer is option 1. Then next one. An antigenic determinant to which a given antibody binds is called. So antigenic determinant, it is known as the epitope. And it is to the epitope that the antibody binds. It is this epitope, it is found on the antigen. Then other options, if you see epimer, it is a molecule that differ in the configuration of, okay, so epimer is that. Cytokine is a signaling molecule found in immune response. Anticodon is the 3 nucleotide sequence found in tRNA molecule, and this anticodon, it is complementary to the codon in the messenger RNA. Then next one, 4-hydroxyproline is an example of non-standard amino acid. It is not a standard amino acid. You know the standard amino acid, there are 20. But hydroxyproline is not a non-standard. It's not a standard amino acid. It is a non-standard amino acid. You know what is a hormone, antibiotic and an antibody. So I am not explaining all that. Then next one. A substance that can act either as a base or as an acid, it is called as an ampholyte. Okay. Then what is an analyte? Analyte is a substance that is being analyzed or measured. Antagonist is a substance that opposes or blocks the action of another substance. And aptamer, it is a short single-stranded DNA or RNA molecule that bind to a target molecule. Then next one, a DNA or amino acid sequence consisting of the residues that most commonly occur at each position in a set of similar sequence. So here what they have asked is, here there are a similar set of sequences. These sequences can be from any organism. Okay, from human beings, from gorillas, from chimpanzees, Okay, or any other primate organism or any organism, if you take, you can find in these sequences there are certain residues that occur commonly at some specific position. Okay, in these sequences there are certain residues that occur commonly. Suppose it is TA sequence that is occurring commonly in all the sequences. Then what is it called? That is what they have asked. So they are called as consensus sequences. Then classified sequence, I don't know whether such a term exists also. Conjugated sequence also I have not come across. Then compound sequence, I don't know what it is. I don't know whether such terms are there. Okay. Then a spatial arrangement of substituent groups that are free to assume different positions in space without breaking any bonds due to the freedom in bond rotation is called. So, spatial arrangement of substituent groups that are free to assume different positions without breakage of bonds. Okay. What it is called? So, it is called as conformation. Because the bonds can rotate. Okay, because these bonds can rotate, they can adopt different shapes or conformations and there is no breakage of the bonds. So that is called as conformation. Then what is a contig? It is used in genomics, that is sequence of DNA that have been assembled from overlapping fragments. It is called as a contig. Conjugation, rotation, you know, conjugation, you know. Then a series of overlapping clones defining an uninterrupted section of a genome is called contig. Then contour I have not come across in genomics. Capsid, it is the viral protein shell. Then chiral, it is found in chemistry. Chiral carbon atoms, like that. And they have non-superimposable mirror images. Then enzymes that create a phosphodiester bond between the 3' end of one DNA segment and the 5' end of another are called ligases. So ligases, they create phosphodiester bond between 3' end of one DNA segment and the 5' end of another. They are called as ligases. And they are involved in DNA replication, then sealing gaps between Okazaki fragments, that is in DNA replication as well as in DNA repair. Then liases, they break various chemical bonds, but they do not involve reactions like hydrolysis and oxidation. Then oxidoreductases, they catalyze oxidation reduction reaction. Hydrolyses are enzymes that catalyze hydrolysis reactions. Then the component of the total energy of a system that can do work at constant temperature and pressure is called. It is called free energy. Okay. Enthalpy, it is the measure of total energy of a system, including internal energy. Okay. Then entropy, it is the measure of the disorder or randomness of a system, and kinetic energy, it is the energy associated with motion. Then the pH of at which an amino acid has no net electric charge and thus does not move in an electric field is called isoelectric or Switzer ionic. So both these terms are correct. It is isoelectric. Then what is isomer? That is molecules with the same molecular formula but with different arrangement of the atoms. Then isoprene, it is the building block of rubber. Then isoenzyme, it is the enzymes that have the same catalytic activity but differ in the sequence of amino acids or their physical properties. Then next one, enzymes that play two distinct roles, at least one of which is catalytic, the other may be catalytic, regulatory or structural are called. Okay, so enzymes that play two distinct roles. So enzyme here have two roles. One is catalytic. The other role of the enzyme may be catalytic or regulatory or structural. Okay, such enzymes are called moonlighting enzymes. So mutases, they catalyze the transfer of functional group from one molecule to another. Polymerases, they help in the addition of polymers. Help in the formation of polymers by adding monomers. Then paralogs, they are genes which have evolved from common ancestor but have distinct function. Then next one. Which of the following statements are correct? DNA microarray is a collection of DNA sequences immobilized on a solid surface that can be probed by hybridization. Yes, that is correct. Then differential centrifugation describes the separation of cell organelles by different rate of sedimentation. Yes, they separate on the base of size and density. That is also correct. Amino acids that cannot be synthesized by humans but are synthesized by microbes are called essential amino acids. Yes, that is partially correct, because amino acids that cannot be synthesized by humans, they are essential amino acids. Of course, they can be synthesized by microbes, but this is not the exact definition of an essential amino acid. Essential amino acids by definition is those that cannot be synthesized by humans and have to be obtained from the diet. They are called as essential amino acids. So C is not correct. Equilibrium constant is specific for a chemical reaction. Yes. That is also correct. So A, B, D are correct. So answer is 1. Then next one. DNA ligases are enzymes that create phosphodiester bond between DNA fragments. We just discussed that. So that's correct. A protein containing one or more prosthetic group is called a conjugated protein. So what is a prosthetic group? It is a non-protein component that is attached to the protein. And a protein that has prosthetic group is a conjugated protein, and a protein without a prosthetic group is an apoprotein. So B statement also is correct. A type of enzyme inhibition reversed by increasing the substrate concentration is called competitive inhibition. That is also correct, because in competitive inhibitor, the inhibitor binds to the active site of the enzyme. Active site is nothing but where your substrate binds. So the inhibitor binds to this active sites and it competes with the substrate for the binding to the active site. Now suppose you increase the concentration of the substrate, you can overcome the inhibition. Okay, now more substrate molecules will be present and the inhibitor will not be able to compete with more substrate molecules. So that is competitive inhibition. So C is correct. Two stereoisomers of a given sugar are that differ only in the configuration about a carbonyl carbon atom are called anomers. That is also correct. So A, B, C, D all are correct. Okay, what is anomeric carbon? It is the C1 carbon in the case of glucose and C2 carbon in the case of fructose. So A, B, C, D are correct. So answer is option 3. Then next one. A researcher engineers a lac operon on a plasmid, inactivating all parts of the lac operator and the lac promoter. Okay, only the lac operator and lac promoter are inactivated, whereas the lac ZYA genes are still active. Now what he did, he replaced the lac operator and the lac promoter with the binding site of the LEXA repressor and the promoter that is regulated by LEXA. So he replaced the lac operator and the lac promoter with LEXA repressor and LEXA and a promoter. The plasmid is introduced into E. coli cells that has lac operon with an inactive lac Z gene. So the plasmid they took and introduced into E. coli cell. So the E. coli cell, it is having a genome there. In the genome, the lac operon is there, but it is having inactive lac Z gene. Okay. Under what condition will these transformed cells produce beta-galactosidase? Now, since the E. coli chromosome, it is inactive lac Z gene, the lac Z beta-galactosidase has to be produced from the plasmid. Understand? Since lac Z gene in the E. coli chromosome is inactive, from where will the bacteria get beta-galactosidase production? It is naturally from the plasmid. I told you in the plasmid only the lac operator and lac promoter are inactive, whereas the lac ZYA genes of the lac operon are active. So beta-galactosidase has to be obtained from the plasmid. Now how is it that possible? So LEXA repressor, what is it doing? What is the action? So LEXA repressor, it is a protein that is found in the SOS response, and SOS response, it is involved and it is activated only during DNA damage. So whenever there is DNA damage caused by UV light, the SOS response get activated. Now in normal condition, that is when there is no DNA damage, the LEXA repressor will be bound to the the LEXA repressor will be bound to the gene for the SOS repair mechanism, and the SOS repair mechanism will not be activated when DNA is not damaged. Okay. Now what happens when DNA damages? So when DNA is damaged, LEXA repressor protein will be degraded. Okay. So what will happen? And the promoter regulated by LEXA will get activated. Now once the promoter is activated, RNA polymerase will bind, and the enzymes and the proteins or the enzymes which are attached next to the promoter region will be transcribing and translating and produce proteins. This is what is happening in SOS response. Now here the LEXA repressor and the promoter have replaced the lac operator and lac promoter, but the proteins which are attached after the LEXA promoter, it is your lac operon proteins. That is your beta-galactosidase, permease, etc. So here Lexer repressor has to be degraded. Now how can that be degraded? That is by using various DNA damaging agents such as UV light. So once the Lexer repressor is degraded, the promoter will get activated and the RNA polymerase will bind to the promoter and it will start synthesizing your beta galactosidase gene. So here if you see first option, on subjecting E. coli cells to high levels of DNA damaging agent like UV light. That is the correct answer. Starving E. coli cells will not do. Growing E. coli cells in lactose rich medium, no, because lac operator promoter are not there. Co-culturing E. coli with bacillus is also not correct. Then next one. Which of the following is the basis for protein structural classification? No, length is not correct. Genus also not correct. Composition also not correct. It is motif. Then next one. The catabolic lysosomal degradation of cellular proteins is called. It is called as autophagy. And what is phagocytosis? It is the process by which cells they will engulf and degrade bacteria or dead cells. That is autophagy. Then chaperones, it is a protein that help in protein folding. Autotroph, it can produce its own food through photosynthesis. Then which one of the following, that is number of molecules per gram molecular weight, represent the Avogadro number? So they have asked what is the Avogadro number. It is 6.02 into 10 raised to 23. You are studying it from your high school. So answer is option 3. Then next one. The energy derived from non-covalent interaction between enzyme and substrate is called. So they have asked non-covalent interaction. So it is binding energy. And this binding energy, it can be through hydrogen bond, electrostatic interaction, van der Waals, hydrophobic interactions. All these are non-covalent interaction. Bond energy is the energy required to break chemical bonds. Activation energy, it is the energy required to initiate a chemical reaction. Entropy is the measure of the disorder or randomness of the system. Then next one. 1 calorie equals how many joules? It is 4.18 joules. Answer is option 2. So thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of biochemistry being discussed, you can go to my channel Omni Bio Entrance. In there you can see for the playlist. In the playlist see for CUET exams and there you will find. Thank you.